Hello. Students, do you know what is mean by enzymes? So first I'll introduce about what is mean by enzymes. Enzymes are acting as a biological catalyst, okay? They will increase your biochemical reaction and they will also do not change the equilibrium constant and they does not change themselves after reaction. You are seeing this line, but what do you mean? They will be, I'm saying in the first point, it is a biological catalyst. Biological catalyst means what is the work of a catalyst? It will try to speed up your reaction, right? The amount of substrate consumed, amount of product released will be same, but it will try to increase your fastness. Okay. Catalyst will speed up the reaction, right? So biological catalyst means they are biologically produced, which will be acting in your biological system to increase the speed of the reaction. Okay, means if uh, two molecules will be converted into two molecules of substrate converted into two molecules of product, the time taken here is 1 hour. But in the presence of enzyme, it will be converted in 1 minute only. In 1 minute, your 2 moles of substrate will be converted into 2 moles of product. Got it? So your enzyme is trying to speed up your reaction and increase the rate of biochemical reaction. Okay. Next, they will do not change the equilibrium constant. Means if you are giving 5 moles of substrate, it will give 5 moles of product only. It will does not change your equilibrium constant also. This will be unchanged after reaction because it is like a teacher explaining a class. If you want to read by yourself, you will take some time, right? It will take literally 3 hours. But when a teacher explains, the problem will be solved in 2 to 3 minutes, right? So in the same way, it will be, but teacher will does not change, right? Even after explaining, she will be in her position only. So this will be unchanged. Enzymes are unchanged even after reaction. They will not be captivated or they will not be consumed in your reaction, and this will bind substrate at the active site. Do you know what is mean by active site? If it is an enzyme, it will have an active site. Active site is the place where your enzyme binds and they will be active site is the site of your enzyme where your substrate will get and attached. Okay. Next, what is its important? Enzymes will speed up reaction without being consumed. That's it. Next is function of the enzyme. What is function of the enzyme? They will control your cellular metabolism. You will take diet, right, which contain carbohydrates, proteins, and lipids, right? They all has to be metabolized, right? Carbohydrates, glycolysis, then Krebs, proteins by proteolysis in your intestine by intestinal enzymes. Then they will be absorbed into your small intestine. Then will be transported and beta fatty acid, beta oxidation of lipids. All are cellular metabolisms, right? They should be carried out by your enzymes only. In the glycolysis, it is a metabolism, right? Metabolic pathway. It require enzymes for protein breakdown or nitrogen bases, nucleic acid synthesis, you require amino acids, right? These amino acids will also be converted by using your enzyme. So, you are saying that they control the cellular metabolism and show substrate specificity. What is meant by substrate specificity? Proteases will act on proteins only. They does not acts on carbohydrates. Okay. If it is a amylase, what is the function of amylase? Do you know what is the function of amylase? It will try to digest your carbohydrates but not your proteins. So, every enzyme has their specificity to digest its substrate. Okay. And they also regulate biochemical pathways and essential for life process. If these enzymes are not present, what will happen? There will be lot of problems in your body. Even due to less amount of your enzymes, your body cannot cope up with the situation. Okay. Next is, what is the characteristics of the enzyme? Characteristic means, if I say Ram is good, you will say characteristics, right? In the same way, enzymes will also have some characteristics. So, mostly your proteins. So, enzymes are made up of proteins. All proteins are enzymes, but all enzymes are not proteins. Why? All proteins are enzymes. But not all enzymes are protein. Why? There is an exception due to the ribosome. Ribosome is an enzyme which is made up of RNA, due to which this expectation come into our picture. So this enzyme will possess 3D structure. Okay, they will be not only in alpha or beta, it will be in the form a 3D structure, like it will be in 3D structure. Got it? Next, even in minute qualities, it will do its work in very great efficiency. Okay, they will show a high efficient catalysis. Then they will be remain unchanged even after reaction, because I already said, if it is substrate, if it is a ma'am explaining you a problem, it will take only 20 minutes. But if a student try to study and understand it, it will take 1 hour. So teacher is a catalyst which is making you to understand in less time. In the same way, enzymes are taking less time to convert it into your substrate into product. Okay, enzymes will convert your substrate into product and they will not be changed even after reaction. Next, activation energy. What is mean by activation energy? It means the minimum amount of energy required for an reaction to start. If I say student to go to school, he will not go, right? I should say him, if you go to school, they will give chocolates, or if you go and come, I'll give chocolate. So I should give some energy to them. In the same way, activation energy is the energy required to initiate a reaction. Only a student go when he get a chocolate. In the same way, the reaction requires some energy to initiate the reaction. Okay. So activation energy will act as a energy barrier, right? A children will always see for chocolate. If chocolate is not there, he will think, no, I'll not go. In the same way. Okay. Enzymes lower the activation energy. Okay. Next, it will increase the reaction rate. What is important? Lower energy. See here. When you see, it is the reaction, right? It is the energy. Here you can see activation energy of uncatalyzed reaction. If there is no enzyme, if he does not have any problem in his tooth, he will eat lot of chocolate, right? If he does not, if he have toothache or something, he will take it only one chocolate. In the same way, here what is happening? If there is an enzyme, the activation energy will decrease. In the absence of enzyme, the activation energy will increase. See, in the presence of enzyme, activation energy decreases. In absence of enzyme, your activation energy decreases. You can see activation energy, uncatalyzed reaction. Uncatalyzed reaction means enzyme does not come into your picture. This is the case. Activation energy is very more. when you compare it to the catalyzed reaction. Catalyzed reaction means enzyme is acting on here. Okay. Next is your coupled reaction. What is mean by coupled reaction? Here you are making an non-spontaneous reaction. What is mean by non-spontaneous? Unfavorable reaction. You are making non-spontaneous reaction into spontaneous reaction, means unfavorable reaction into favorable. Okay. See here, it is coupled reaction means it is coupling anabolic reaction and catabolic reaction. Catabolic reaction will release energy, anabolic reaction require energy. First, what is mean by catabolism? What is mean by anabolism? Anabolism means energy building, right? From small molecules, large molecules will be produced. Coming to catabolism, large molecules will be broken into small molecules. When you try to build a big house with small bricks, you will require lot of money and lot of constructors to work on it, right? So anabolism require lot of energy to make your molecule large with the small molecules. Catabolism is just breaking large molecule into small. It is very easy, right? So when you make large molecules to small molecules, energy will be released. But when you convert your small molecule to large molecule, you require some energy. Okay? For building and for constructing and building, you require money from your relatives. But to break your building, you does not require much money. Even if you give some money, they will come and they will just demolish easily. Okay. So they both will be linked through ATP and maintain cellular energy balance. See here. If you see conversion of glucose to glucose 6 phosphate is an hard reaction. So what it is happening here? In this conversion, what you are using, you are breaking your ATP and converting it into ADP and inorganic phosphate to release some energy. What is the enzyme here? Do you remember enzyme? What is the enzyme in the glucometabolism? You already have studied in your inter also. Glucose to glucose 6 phosphate. What is the enzyme? Hexokinase. Okay. So it is one reaction, it is second reaction. Here what is happening? You are using an favorable reaction to make unfavorable reaction to favorable. Ante enti? You are making a non-spontaneous reaction into spontaneous reaction by linking them. Means one is studying good, another is not studying good. You are linking the person who is not studying with the person who is studying. So what will happen? For 2 days he will neglect, but after third day he is also studying, so I will also study. It is the example for it. Okay, next. Then I little bit explain you about what is exergonic reaction, what is endergonic reaction. Okay. First, coming to your exergonic reaction require energy. It require energy. Okay. Coming to your endergonic reaction, it will release energy. If it is releasing energy, what this process is? Any catabolism. Endergonic reactions are catabolic reaction, where exergonic reactions are anabolic reactions. Okay. Next, they are spontaneous. Exergonic reactions are spontaneous reaction. Endergonic reactions are non-spontaneous. Next is we will see about the classification of enzyme. Here, how many classes? 1, 2, 3, 4, 5, 6. 6 classes were there, right? They normally your enzymes will end with A's. Now we will see about the different enzymes. First is oxidoreductase, transferases, hydrolases, lyases, isomerases, ligases. So enzymes are divided into six major class based on your IUB. Okay, they are oxidoreductase, transferases, hydrolases, ligases, isomerases, and lyases. So first coming to your oxidoreductase. Oxidoreductase. What it is? It will catalyze your oxidation reaction and also reduction reaction. They will transfer the electrons or your hydrogen atoms. Okay, they will do oxidation or reduction. They will transfer your hydrogen or your electrons. Okay, got it? Next, example is dehydrogenase. Next is transferases. Transferases means they are trying to transfer any functional group like amino group or phosphate group, etc. See here, transfer of group of atoms from one molecule to another molecule. What are the molecules like amino group, phosphate group, etc., etc. So here what is happening? A, B, and C. Here, what is happening? B is getting transferred to your C. This is the reaction. Got it? Next, what is happening? What is the enzyme? Transaminases, phosphotransferases, and hexokinases, transaminases. They all comes under your transferases. What is your example? If I say, I already said you there at the time. Glucose with the ATP. What is happening? Glucose 6 phosphate and ADP. What is happening here? One phosphate which is present in the ATP is getting transferred to your glucose, due to which your glucose will be converted into glucose 6 phosphate, right? So what is the enzyme working here? Hexokinase. Next, coming to hydrolases. Hydrolases, what is the function of hydrolases? They will break molecule using water. Okay. Hydrolysis of the substrate by addition of the water molecule. Here see, if it is an AB is one substrate, here you are using water. Water will be broken into hydrogen and also OH minus, right? So what is happening? Your hydrogen is getting and bound with your substrate A and your OH is binding with B molecule. So what it is happening? The bond between A and B is broken. A is settling with the hydrogen atom where B is settling with the OH atom. Example, all your digestive enzymes like amylases, pepsin, trypsin and chymotrypsin, everything. There are some enzyme amylases, all digestive amylase enzymes will directly use the water molecule to break your bond. I already said all the biomolecules are linked by the bonds, right? They will be formed due to release of water. They are condensation reactions. So there are only two reactions for bond. One is making bond by condensation reaction and breaking of bond by hydrolysis reaction. Okay. Next, isomerases. What is the function of isomerases? So they will convert one isomer into another. Example, phosphoglucomutase. Okay. So first coming to isomerases. This will control the conversion of one isomer to another by transferring a group of atom from one molecule to another. See here. If it is an A, B, C, it is just changing the position, right? Isomer means if I say change in only one position, the empirical formula isomer, what is mean by isomer? What is mean by isomer? Isomer means having same empirical formula. If it is C6H12O6, glucose and fructose are isomers, right? So they have the same empirical formula but different arrangement. See here, this compound contain ABC only, but the arrangement is different. So what is the enzymes are called? Isomerases. Okay. Next, liases. Liases means they can add or remove groups without water. Okay. they can add or remove the carbon dioxide. So they will break the chemical bond without addition of the water. They can add another molecules like carbon dioxide. So A and B can become A plus B. Decarboxylases and carboxylases all comes under here. Coming to ligases. What is meant by ligase? Ligase in DNA, the Okazaki fragments are bound by, or the gap is filled, or the nick is sealed by your ligases, right? So formation of new chemical bond using ATP as a source of energy. See here, DNA ligase helps in attachment of your Okazaki fragments. See here, A plus B, by using ATP, what is happening? The bond between them is created due to ligases. If you see, this is a different fragment, this is a different fragment. By breaking off ATP, by using some energy, there is a bond created by A and B. What is the enzyme here? Ligases. Next, we are going to study about the cofactor. What is meant by cofactor? They are non-protein components required for enzyme activity. Got it? So, you have to learn some terms like apoenzyme. I'll write here. Wait. Apoenzyme. What is meant by apoenzyme? It is a protein part. Okay. Apoenzyme is a protein part. Coming to cofactors. What is meant by cofactor? They are non-protein part. Apoenzyme is a protein part where cofactor is a non-protein part. Coming to holoenzyme. What is meant by holoenzyme? Apoenzyme plus cofactor is known as holoenzyme. Holo means full formation, right? So first coming to apoenzyme, it is a protein part where cofactors are non-protein. Non-protein parts, when bind with your protein parts, your enzyme become functional sometimes. So enzyme requires some cofactors for their work. Okay. So there are some types of cofactors. In the slide I mentioned prosthetic group, coenzyme, metal ions. What is meant by prosthetic group? It will be permanently attached. Permanent attachment. Example, heme. Okay, for your hemoglobin, your heme will be attached to your globin permanently. Once heme get attached to your globin, it does not lose. Okay. Next, coming to your coenzymes. They are loosely attached. What is your example? NAD plus and CoA, coenzymes. They are acetyl-CoA. When you see acetyl-CoA, one is a protein part. CoA is a cofactor, right? So they both combine to do a function. Coming to metal ions, you already know magnesium, zinc, copper, manganese, all comes under your metal ions. Okay. Next, example, your zinc. For carboxypeptidases, if zinc is not there, they cannot perform their function. Coming to, I already said, right, glucose will be converted into glucose 6 phosphate by the hexokinase, right? If this hexokinase has to do its function, it require a cofactor called magnesium. Okay, if the magnesium is not there, it cannot work. So now we will see about the mechanism of the enzyme action. You can just normally say enzyme will combine with the substrate. It will give a enzyme will combine with substrate to give a enzyme substrate complex, which is a transition state. Okay. Now after this you will be getting product, right? But it will be producing like enzyme product. But after the reaction, I said that even when substrate is converted into product, your enzyme does not get eaten up, right? It does not consumed. It will be freely again released to do the another reaction. The same way, see here, first, in the first state, how was it? In the last state, it is also same. Okay. So first, enzyme will bind with your substrate by forming, it will form a enzyme substrate complex, and your enzyme product will be formed, enzyme will be released, product will be released. So our first step is binding. Your second step is catalysis. Third step is product release. Okay. Next, you have to know one important thing, active site. It is called as active site. I said that active site is very specific for substrate binding region. This will act as catalysis region and that will determine the specificity. This active site is formed due to the protein folding. Okay. Next, I said that enzyme specificity, right? Enzyme specificity means absolute group specificity or sometimes linkage specificity. Example, when you see a urease, uh, example, hexokinase. Hexokinase is very specific to the group, where chymotrypsin is very specific to your linkage. Got it? Next, proteolytic enzymes. When you see subtilin, it can cleave any peptide bond. Trypsin will cleave only after lysine or arginine. Coming to thrombin, it is also very specific for the peptide bond. Got it? Next, we will see about the lock and key mechanism. Do you know who proposed this lock and key mechanism? It is Emil Fischer. Okay. So he said that active site is rigid and substrate fit perfectly. Okay. See here. Why we gave the lock and key mechanism? See here. The substrate and your enzyme. I said that enzyme is very specific to substrate. If the enzyme is in round shape, if the substrate is in this shape, enzyme key lock and key model does not get applicable. Okay, see here. It is a substrate, it is a lock, which is we called as enzyme. So lock and key complex, enzyme substrate complex. Okay, next, induced fit theory. Induced fit theory means here what they are saying in first we said that enzyme active site is fixed. Enzyme active site does not change for the substrate. But coming to here, based on the substrate, the enzyme will try to change itself or modify itself to release your product. Okay. This is known as induced fit theory, which is proposed by Daniel Koshland. Okay. Next, coming to your molecular basis of your enzyme action. Mostly it will be having 3D structure. You can see here, right? It will be in a 3D structure. And active site is found by the folding of the protein, right? And amino acid chains will also participate. Hydrogen bonds will try to stabilize your substrate. Okay. Next, what is your mechanism of catalysis? Okay, I'll say here. Hydrophobic environment. What is the factors responsible for your catalysis? There should be a hydrophobic environment and acid-base catalysis and also covalent catalysis. Got it? Next, they all helps in lowering your activation energy. Next, what are the factors affecting your enzyme action? What do you mean by factors? Enzymes are proteins, right? Proteins can work at medium temperature, but they will get broken or denatured in the high temperature. So there are some effects of temperature. So first is effect of temperature. Next we will be seeing effect of pH and enzyme concentration. Next allosteric site and allosteric enzyme and isoenzyme. First coming to effect of temperature. Rate will increase with temperature, but maximum at the optimum temperature it will work. But at high temperature it will denature, which means the enzyme can work at optimum pH. But once the temperature is very high, the denature of your proteins will take place. Okay, denaturing of protein takes place at high temperature. The all enzymes will have optimum temperature. Okay. Next, maximum temperature. Normally, optimum will be 37 degrees centigrade. Okay. Next is effect of your pH. Every enzyme has optimum pH. Extreme pH can also cause denaturation. Pepsin. Mostly all enzyme will be in neutral form or little bit acidic. It is okay. But pepsin optimum pH is 2, which means it is present in your stomach, right? So even in the HCl condition, it has to work, due to which it try to develop a very resistant pH, which is acidic pH 2. Okay. Next, your rate directly proportional to the enzyme concentration. Keep it in the mind. Next, coming to allosteric enzymes and isoenzyme. What is mean by allosteric enzyme? Which means active site and regulatory site. They will be get activated or inhibited by the effectors. They will show a cooperativity. Okay. Next, active site means which will increase your action. Regulatory means which will stop your action. So allosteric enzyme is having both the sites. Okay. Next is isoenzyme. They will be different forms of same enzyme. They will catalyze the same reaction. Example, lactate dehydrogenase. Okay. Next is your enzyme kinetics. Do you know why do you study enzyme kinetics? To study the rate of enzyme catalyzed reaction. That will help you understand enzyme mechanism and describe how your reaction rates are affected by substrate concentration, enzyme concentration, temperature, pH inhibitors, and also your activators. So, it will mostly help you to know how enzyme function inside the cell. Okay, you cannot go into the cell and you cannot see, right? So, enzyme kinetics are your physical proofs to study your enzyme. Okay, next, coming to measurement of your enzyme activity. How you will measure it? First, enzymes will be incubated with the substrate under optimal condition, and the activity will be measured by disappearance of your substance and appearance of product. See, I gave a good nutrition agar in a petri plate. I left it for air for 1 hour. Now what will happen? You will see growth of the fungal. If it is a potato agar solution, you can see the fungal growth. Here, your nutrition agar is completed and you can see the fungus. So what is happening? Disappearance of substrate and appearance of your product. So this sample will be analyzed at regular intervals. Example, conversion of your NAD plus to NADH, and NADH absorb light at 340 nanometers. So what you will do, you will prepare your NAD solution and you will prepare a solution which will contain NAD plus, and at different intervals after 10 minutes you will absorb like under 340 nanometers in colorimetry, 20 degrees centigrade, 30 degrees centigrade, 40 degrees centigrade. At certain degree of centigrades you can see its absorption is very high, where you can say that NAD is converted into NADH. Okay. Next, saturation of the enzyme. What do you mean by saturation of the enzyme? Do you have any idea? If I give, how to say, I'll give you 10 molecules of substrate and 5 molecules of enzyme, they will directly give 10 molecules of product, right? Here, now what I'll do? Enzyme concentration, I'll keep it constant and I'll increase the substrate concentration and increases the reaction rate. At high substrate concentration, rate become constant, all the active sites become occupied, right? Due to which what will happen? If it is only 5 molecule, I give 100 molecules. Rate of reaction, I'll give you only 5 minutes of time to convert everything into your product. In 5 minutes, I can convert your 10 moles, but As you increase your substrate, what will happening? The enzyme, all the enzyme active sites been occupied by your substrate. Now this enzyme has to be converted into product, and again this enzyme has to go to catch up your substrate. Now what will happen? It will become saturation. The substrate will be saturated. Okay. So initially many free active sites will be available, which means when there are 10 molecules, you are giving at the first 1 minute, what will happen? This enzyme, all 5 active sites will be very free and available. But as more substrate increase, more enzyme substrate complex will be formed. Eventually, enzyme becomes saturated, which means all enzyme got their work. Now, none of them are free to do the new work. So, further increase in the substrate has no effect. What it is here, saturation occurs because enzyme concentration is limited. But if you also increase the enzyme concentration, you can increase your product. Okay. Next is your chapter 2, bioenergetics. Did you remember how much today you studied? And I forgot to say something. Inhibition. Enzyme get inhibited at extreme temperature and extreme pH due to alcohol, acetone, and specific inhibitor. And there are two major competitive types. First is competitive inhibition, another is non-competitive inhibition. First, coming to your competitive inhibition, what will happen here? The inhibitors will resemble your substrate, due to which in the active site of your enzyme, your inhibitor will go and bind, due to which your product will does not form. Coming to non-competitive inhibitor, what they will do? This non-competitive inhibitor will bind at other than the active site. When it bind here, what will happen? Your active site which is present will change. If your substrate has this activity, if it is lock and key mechanism, it does not work, right? Because your substrate structure is different. Your enzyme active structure is different, due to which what will happen? It cannot be converted into product. So no, competitive inhibitor will attach at active site, non-competitive will bind at other than the active site, due to which what will happen? Your product will does not form. So coming to examples of competitive inhibitor, when you take sulfanilamide. So it will compete with your PABA that will block your folic acid synthesis and also inhibit your bacterial growth. Okay. Next, coming to your non-competitive thing, I already said they will bind other than the active site, that will change the enzyme structure, that will make the enzyme inactive or less active. They cannot be overcome by excessive, even if you give high concentration of substance, that will does not affect your substrate binding. Got it? Next is your second chapter called bioenergetics. Do you know what is mean by bioenergetics? Students, if you don't respond, how can I say? So coming to your bioenergetics, what is meant by bioenergetics? It is the study of energy transformation in the living organism, including your energy production, storage, transfer, and utilization. Okay. Bioenergetics means here you will try to study how your energy is transferred from one body to another body, and how this energy is created or lost. You know thermodynamic laws, right? All thermodynamics law you will be studying here, and you will study about the entropy, enthalpy, Gibbs free energy in your bioenergetics. I'll be explaining very brief about it. So try to see by yourself again, so you will understand nicely. So coming to function. What is the function? The energy generation from the nutrients. They will store of energy as ATP, and also utilization of energy for cellular work, and study of biochemical reaction using thermodynamic principle. Next, see. First coming to thermodynamics. What is meant by thermodynamics? Thermodynamics means you are trying to study the system. System means which is present in your universe. Okay. There are three types of systems. Okay. First is open system. Second is closed system. Third is isolated system. First, coming to open system, first you have to remember where there is a matter exchange, where is energy exchange. Okay. First, in open system, what will happen? The matter will get exchanged and also energy get exchanged. But coming to your closed system, uh, human being is an open system. We will exchange matter, which means we will take the food, we will remove the fecal matter from our body, and we will absorb some energy, right? So matter will be exchanged. Environment is giving us, we will giving something to the environment, and we are producing energy to work, right? So coming to closed system, when you take a bottle, flask, okay, if you keep hot water, what will happen here? The matter will does not get released to the outside, but the energy will be released. Even when you touch your flask, you can heat little bit of heat. Heat is known as energy. Okay. In isolated system, you cannot see the matter exchange or you cannot see the energy exchange. Okay. Next is your first law of thermodynamics. What do you mean by first law of thermodynamics? Any idea? It is also known as law of conservation of energy. You can say that energy can be neither created nor lost, but it can be transferred from one body to another body or one form to another form. Here, what is the equation? Delta E equals to Q minus W. Delta E means change in internal energy. Q means heat absorbed, work done. Here, when I give some energy, okay, to do some work, it will absorb some energy, right? So the energy, if you want to know how much energy is changed, I gave 100. Okay. I want to know how much energy is lost. Now, if I do a work which cost 80, okay, I'm trying to absorb 80 kilojoules of energy from a system. But a work is done with 8 kilojoules, right? Remaining 20 kilojoules will be left out. It can be used to do the another function, right? So this 8 kilojoules helped you to complete your work. Work is done. This work is utilized to do the another work, right? So your energy is converted into one form to another form, but it does not get changed. Next, what is its significance? The total energy of remains will always constant, right? If you eat a apple, you are gaining some energy, right? This energy will be utilized to do some work like planting. So what it is happening? It is helping your future generations. But energy is neither lost nor created. This will explain your energy transformation in the living organism. Next, we will see about the enthalpy. Okay. Uh, Enthalpy, if you have the enthalpy negative, it is known as exothermic reaction. Exothermic reaction means energy released. If delta H is in positive, it is an endothermic. Energy is consumed. Ma'am, why are you saying enthalpy? You will know while you are studying Gibbs free energy. Okay. First, you try to remember that it is a heat energy. Got it? Next, negative means it is trying to release the energy. If it is in positive, it is trying to consume the energy. Remember, if delta H is negative, exothermic reaction. Negative people always try to give the rumors. Positive people will always try to keep it themselves. Okay? It is for delta H, enthalpy. Enthalpy delta H, negative energy will be released, exothermic reaction, where if it is a positive, it is an endothermic reaction, energy will be consumed. Example, glucose oxidation is a highly exothermic reaction. Next, your second law of thermodynamics, right? What is meant by second law of thermodynamics? Every spontaneous process increase the entropy of the universe. Entropy means it is the measure of randomness or disorder. If you keep an apple on plate for 10 days, it is okay. But after 20 to 30 days, it will get spoiled and the surface area will decrease, which is known as entropy, randomness. So after 100 days, what it will be? It will be degraded into powder. It will become a fungal form. So the plate which you are using totally occupied by your spoiled apple. So randomness of your system is increasing. Okay. Next, you will see now what is mean by entropy and enthalpy. Entropy is randomness of the system. Enthalpy means heat energy. Okay. See here. What is mean by Gibbs free energy? Delta G equals to delta H minus T delta S. Gibbs free energy. Next, heat is enthalpy change, T is temperature, S is entropy change. Okay. Next, what is the significance of delta G? By delta G value, if delta G is positive or negative, we can see the exergonic reaction, endergonic reaction. Okay. If delta G is less than 0, it is known as exergonic reaction, which means it will release the energy and spontaneous reaction. It is very possible. But if delta G is greater than 0, it will require some energy which is non-spontaneous. Example, protein synthesis and DNA synthesis. Next, another is equilibrium reaction where delta G equals to 0. Okay. Then this reaction is known as equilibrium. And you also have to study the standard free energy change. What is the relation with the equilibrium constant? I'll just say the formula. Delta G naught equals to minus RT in K equilibrium. Okay. If delta G is greater than 1, it is known as negative. If delta G is greater than 1, it is known as positive. If delta G naught is 1, it is known as zero reaction. Okay. This all three cases about the, you have to study the cases in this chapter. Okay. It will be used to calculate the energy released during the electron transfer. Okay. Next, you will also study about the electron transport chain and formation of ATP. You can study by yourself. Next, what is the time? Okay, 2:52. I have to complete more 2 chapters. Okay. Water and mineral metabolism. What do you know about the water? You always drink, right? So you should say little bit. Okay. Water and minerals are essential inorganic compounds of our body. Mostly our body is made up of 60 to 70% of water, right? If there is no water, it will lead to dehydration of your body. So minerals are required for normal metabolic and physiological function that will maintain your cellular homeostasis, enzyme activity and electrolyte balance. So if there is a disturbance in your level, means in your water or your minerals, what will happen? It will dehydrate your body, edema, electrolyte imbalance and acid base disorders. If there is acid base disorders, there are lot of problems in your body. Next, what is the significance? Okay, you should also know when you are studying something, you have to know its uses, its problems, how it also absorbed. First, it is a universal solvent of our body. Next, medium for all biological reactions that will transport nutrition, hormones, gases, and waste. That will also help in regulation of your body temperature, maintain cell shape and volume, and also participate in hydrolysis and metabolic reaction. Next is your body water compartments. First, see what is your total body water. Mostly it will be from 60 to 70% of your body weight, right? So there will be compartmentalizations like two types, intercellular fluid and extracellular fluid. First, coming to intracellular fluid, they will be about 2 to 3 of your body, of your total body water, okay, which means present in. So internal fluids will contain rich in potassium, magnesium, phosphate, and proteins. They are the site for the cellular metabolism. Coming to your extracellular fluid, it will be one third of your total body volume. Okay. Next, that will include plasma and interstitial fluid. That will be rich in sodium and also calcium. Keep it in mind that your body will contain 60 to 70% of water, where the intracellular fluid will be 2 by third of total body volume water, and extracellular will be 1 by third of your total body water. And also try to remember what are the elements mostly present in your compartment. Next, I said that water as a universal solvent, right? So water is a polar and form hydrogen bonds. Due to the hydrogen bonds only, your DNA is stable, right? The nitrogen base pairs, it is your DNA. Due to your hydrogen base pairs between your nitrogen bases, they are getting attached. So they will dissolve the electrolytes, sugar, amino acids, and vitamins. They will facilitate the enzyme substrate interaction, promote dissociation of salts into ion, and also essential for nerve conduction and muscle contraction. Next, regulation of water balance. How it will be regulated? uh, some people don't drink the water. Maybe their hypothalamus is does not working properly. So once visit the hospital. So major regulator mechanisms. What it is? First is thirst center. When you lost a loss of water, when you go into sunlight, after coming home, you will always feel thirst to drink some water. Even when you take masala foods, you will always try to take more water to balance your body, right? So, and AD, anti-diuretic hormone, and aldosterone, and your kidney. They are the regulatory mechanisms of your water balance in your body. Then, what is its function? They will maintain the blood volume. If water content is less, your blood will become very thick. Okay, their nature will become like a jelly like nature, due to which your circulation will does not take place properly in the body. Next, they will also regulate osmotic pressure and control water reabsorption and excretion, vasodilating hormone. Okay. Next, here you will be studying about the re- regulation of water balance, water as a universal solvent. Next, I'll say little bit about you dehydration. What is mean by dehydration? Dehydration means loss of water. Okay. Next, this is caused due to diarrhea, vomitings, excess sweating, or due to fever. Sometimes diabetic person will lose lot of water due to diabetes mellitus and diabetes insipidus. That will reduce your body volume, electrolyte imbalance, and also impaired metabolism. So there are different types of dehydration, like isotonic dehydration, then hypotonic dehydration, and also your hypertonic dehydration. Okay, so Coming to isotonic dehydration, it is like equal loss of water and electrolytes. Your water and your electrolytes are equally lost. Coming to hypotonic dehydration, here electrolytes will be greatly lost and your cells will swell. Okay. Coming to your hypertonic dehydration, here your water loss is more and cell will shrink. So, first we studied about the dehydration. It will be caused due to diarrhea, vomitings, and sugar patient, diabetes mellitus, and diabetes insipidus. And what will the effect here? Your body volume will be reduced, electrolyte imbalance, and impaired metabolism. Next, what is types of dehydration? There are three types. That is isotonic dehydration, hypotonic dehydration, hypertonic dehydration. In isotonic dehydrations, your water and electrolytes are equally lost. Coming to your hypotonic, electrolytes are most lost. Hypertonic, water is most lost. Hypo, waterless. Hyper, water more. Keep only one thing in mind. Try to remember your water. If water is less lost, hypotonic. If water is most lost, hypertonic. Okay, and water and electrolytes are inversely proportional. Got it? For only these two, you try to remember in this structure. Okay. Next, what is the biochemical consequences due to which your increased plasma osmolarity? Okay. Increased plasma osmolarity, reduced enzyme efficiency, decreased ATP production, impaired glycolysis and TCA. You know that TCA, glycolysis is also called as water releasing cycle. Water releasing pathway. It is not a cycle, sorry. Your Krebs is a cycle, but your glycolysis is a pathway. Next, metabolic acidosis and reduced tissue perfusion. Next, coming to edema. What do you know about the edema? Excess accumulation of fluids in the intestinal tissue. Okay. Excess accumulation of fluids in interstitial tissue. Not intestine, interstitial tissue. What it will happen, due to which it leads to heart failure, liver cirrhosis, kidney disease, malnutrition, and also inflammation. What is the consequences of it? Here, the tissue will be swelling and your reduced oxygen delivery and also impaired metabolism. Okay. Next is your mechanism of edema. How this edema will increase your body under pressure. Okay. So, increased capillary hydrostatic pressure will decrease your plasma oncotic pressure, due to which your capillary permeability will be increased and lymphatic obstruction and sodium water retention will be taking place. Next, you will be studying about your calcium and phosphate metabolism. So first, you have to know where this calcium and phosphate is present. In your ATP, your phosphate is present, right? ATP is the energy currency of your cell. Next, calcium will be present in bone, teeth formation. For muscle contraction, you require calcium. And for nerve transmission, you require calcium. Blood clotting, calcium play very important role. In energy metabolism also, calcium and also phosphate play very important role. First, its storage. Coming to storage, 99% of it is stored in your bones and teeth. 80 to 90% phosphate in bones. See, 99% of your calcium is present in bones and also in teeth. But only 85 to 90% of phosphate is present in bones. Okay. Next, what is the source of your calcium? Milk, dairy products, green leafy vegetables, nuts and seeds, fishes with edible bones. Okay. Next, how it will be absorbed? It can be absorbed by direct transport or passive diffusion. Direct transport means um, vitamin D helps in absorption of the calcium. I said that in the class, during the class, uh, A pregnant lady is prescribed with tablets having calcium and also vitamin D, because vitamin D indirectly helps in absorption of a calcium into your bones or body. Okay. Next, functions. What is the function of calcium and what is the function of phosphate? First, calcium helps in bone mineralization and muscle contraction, neurotransmitter release, blood coagulation, cell signaling, and membrane stability, right? Coming to phosphate, it is a component of ATP. You all know this. And also for DNA RNA synthesis, we require ATP, and also phospholipid formation, enzyme regulation by phosphorylation, acid base buffering. These are the functions of your calcium and also functions of your phosphate. Next, coming to your hormonal regulation of calcium and phosphate. First, parathyroid hormone will increase your blood calcium and stimulate bone reabsorption. Okay. Vitamin D, which is also called as calcitriol, that will increase the intestinal absorption of calcium and phosphate. Okay. Calcitonin, that will lower the blood calcium and inhibit osteoclast activity. Next, what are the disorders of calcium potassium metabolism? So, if there is a no proper calcium or potassium, what will happen? If it is a hypocalcemia, less calcium, it will lead to tetany and muscle spasms. If it is a hypercalcemia, that will lead to kidney stones and bone mineralization. Hypercalcemia means less calcium, hyper means more. Vitamin deficiency. If there is no if there is no vitamins, what will happen? Calcium absorption does not take place. So, if there is a vitamin D deficiency, it will lead to rickets and osteomalacia. Next, osteoporosis, that will reduce the bone mass. Okay. Next is your iodine metabolism. What is the essentiality to study it? It is essential for thyroid hormone synthesis and regulate T3 and T4 production. Do you know what is T3 and T4? It is the cells to confirm your thyroid is there in your body or not. Next, regulates metabolism and growth. What is the source? Ionized salt, seafood, eggs, and also dairy products. Next is your thyroid synthesis. Here, first iodine which is consumed will be taken by your thyroid glands. You know where thyroid glands are situated, right? So your oxidation of iodine will take place. Next, organification of your thyrosin. Next, due to which they both will form MIT and DIT. If two DITs combine, they will form T4 cells. If one MIT and one DIT form, it helps in formation of T3 cells. These cells will be released into your blood. Okay. Next is your acid base balance. Normally your blood pH will be 7.35 to 7.45, which is neutral. Okay. Due to this neutral, buffer system is regulated, your lungs will get good health, your kidney will does not become acid condition or base condition. If there is no proper acid base balance in your body, it will lead to acidosis or alkalosis. Acidosis means pH decreased, which means acid in condition 1 to 7. Alkalosis, pH increases, which means base from 8 to 14. Okay. Next, your fourth chapter called vitamins metabolism. What do you know about the vitamins? They are organic micronutrients which are present in very small amount, but they have very impact on your body. They does not provide energy but regulate the metabolic process. Their deficiency can cause specific diseases. Example, Rickets is caused due to psoriasis, sorry, vitamin C, and also berry all will comes. I'll explain you in next. Okay, these vitamins are classified into fat soluble vitamins, water soluble vitamins. Fat soluble vitamins A, D, E, K, where water soluble vitamins are B complex vitamins B1, B2, B3, B6, B12, everything, and vitamin C. They will be absorbed with dietary fats because they are fat soluble vitamins, right? They will be stored in liver and adipose tissue, and excess intake may causes toxicity. If you take much, it will cause toxicity. Due to this, you have to always take it in a balanced amount. So, coming to water soluble vitamins, they will be water soluble, right? So, they can go out with your water due to your urine, they can go out. So, water soluble vitamins has to be taken again and again into your body. They will be absorbed directly into the blood, but limited storage except vitamin B12 and excess excreted in the urine. Okay, next, what is the general features of your vitamin metabolism? First, they will be absorbed in the small intestine, transport through blood and lymph, the activation into biological active compounds, storage in the tissue, utilization in the metabolic reaction, excretion of excess vitamins. Okay. Next, first, today we will study about the fat soluble vitamins. Coming to water soluble vitamins, I'll explain you three vitamins of D. So you can go through it because we have only 1.5 hours. First, coming to source. In animal, you can see in liver, fish, oil, milk, butter, egg, yolk, and cheese. In plant sources, if it is for non-vegetarians, you can see this. If it is for vegetarians, it is beta carotene, alpha carotene, beta cryptoxanthin, and green leafy vegetables, carrot and mango. Okay. Next, first we will see about vitamin A. Vitamin A will be transported into, first it will be in the form of retinol, they will be converted into retinol ester, then converted into chylomicron, they will be brought into liver through lymph and blood. Okay, liver will release this retinol bound to the retinol binding protein and TTR. Okay, transthyretin. Next, coming to metabolism, retinol will be converted into retinol, which is reversible, but retinol will be converted into retinoic acid, which is irreversible. Next, we will see about the visual cycle. Do you know what is mean by visual cycle? Any idea? First, it will be in the form of 11-cis retinol. This retinol will combine with opsin to form rhodopsin, visual pigment to see your brightness, or when you go out you can see colors or everything, right? Due to which the factor is rhodopsin. Okay, due to light, this rhodopsin, which is known as 11-cis retinol, will be converted into all trans retinol. This all trans retinol will again converted into all trans retinol. See here, it is converted into all trans retinol, right? It first they are photoreceptors, 11-cis retinol and 11 trans retinol. Now, what is happening in the retinal epithelium? This trans retinol can convert into cisretinol, or in the blood it can travel, it can reach the liver. In the liver, what will happen? This trans retinol, by using NADH, it will be reducing and forming trans retinol. This retinol can be converted into cisretinol by isomerase, which is present in the liver. So it is taken in the liver. Keep it in mind. Then this NAD will be converted into NADH to form cisretinol. Retinol NAL is converted into NOL, then NOL is converted into again NAL. This will again convert into 11-cis retinol. Okay. So what is its function? It helps in vision and also cell differentiation, immunity, and embryonic development. Okay. vision, cell differentiation, epithelial integrity, immunity, embryonic development. And they will be mostly stored in the liver, hepatosteatal cells. Next, they will be excreted in bile and urine as water soluble metabolites. Okay. Next is vitamin D metabolism. We say that please go to sunlight so you will get the vitamin D properly. So sunlight, fish, liver, oil, fatty acids or fatty fish, egg yogurt, butter, fortified food contain more vitamin D. And your skin also synthesize your vitamin D. Do you know how it is? Now we will see here. Here, when sunlight or UVB comes onto their skin, your skin will contain 7-dehydrocholesterol. This 7-dehydrocholesterol will be converted into cholecalciferol, which is known as vitamin D3. This vitamin D3, when gone into the liver due to the enzyme 25 hydrolase, this will be converting your cholecalciferol into 25 hydroxy vitamin D3. Okay. Next, they will be also called as calcidiol. Okay. Now, this will be entering into your kidney. In the kidney, your 25 hydroxy vitamin D3 will be converted into 1,25 dihydroxy vitamin D3, which is the biological active form of your vitamin D. Okay. What is the function? I already said vitamin D helps in absorption of calcium, which is given to your uh, delivered mothers. Okay. Increased calcium absorption and also increase phosphate absorption, promote bone mineralization, maintain calcium homeostasis. Coming to your regulation, that will lower your calcium, due to which your pHT will increase, then increased calcitriol synthesis. And if it is high calcium and high phosphate, reduced calcitriol synthesis will take place. Next, coming to your vitamin E metabolism. So, what is the source? Vegetable oil, nuts, seeds, cereals are the source for your vitamin E. First, how they will be transported? First, a small micelles will be created in your intestine. From the intestine, they will be converted into chylomicrons. The chylomicrons can easily enter into your liver, where your very low density lipids, low density lipid, high density lipids will be converted. They will be absorbed by your tissue. Okay. Next, what is function? They will be functioning as antioxidant protection of your membrane lipids and prevent lipid peroxidation. Next, how it will be stored? It will be stored in liver, adipose tissue and skeletal muscles. They will be excreted as bile, feces and urine. Next, coming to your vitamin K metabolism. What is the source here? Green leafy vegetables, you already know, vegetable oil, intestinal bacteria. Next, they same function, same like vitamin E. Chylomicron, liver, VLDL, LDL, DHDL and tissues. What is their function? Activation of clotting factor. You know that vitamin K play very important role in conversion of your clotting factor or activation of your blood clotting factors. Okay. And also bone protein activation. Coming to storage, they will be stored in liver, adipose tissue. Okay. Next, we will see about vitamin K cycle now. So here what is happening? It is required for activation of clotting factor. I said that they helps in blood clotting factors, right? So first what will happen? Vitamin K quinine, which is come from the diet, will be converted into vitamin K hydroquinone by vitamin K quinone reductase. Due to the reduction activity, what is happening? Quinone is becoming hydroquinone. It is the active part. Next, vitamin K hydroquinone will be converted into epoxide, vitamin K epoxide, due to which Gamma glutamate carboxylase helps in conversion of your hydroquinone into epoxide. Now what will happen? This epoxide will helps in conversion of your vitamin K this will be again converted into vitamin K quinone, but there is an inhibitor called warfarin that helps in conversion stopping of your converting epoxide into quinine. Okay, and it is a cycle, right? So it will be repeated. Next is, what is the importance? What is the importance of it? It will activate the prothrombin and also factors like 7, 9, 10, and also for bone proteins. Okay. Next, coming to metabolism of water soluble vitamins. So, water soluble vitamins means they will be absorbed into portal circulation, activated in the liver, tissue, and limited storage excess excretedly in urine. Okay. Next, what are examples? Vitamin B complexes and vitamin C. Now, we will see about first, vitamin B1. First, where it will be absorbed? It will be absorbed in the duodenum, jejunum by active transport, and they will be activated by when thiamine is converted into thiamine pyrophosphate. Okay, how it will be? What are the functions? Carbohydrate metabolism and oxidative decarboxylation reaction. Okay, next, coming to storage. How it will be stored? It will be stored in liver, heart, brain, kidney, and also skeletal muscles. But if there is no vitamin B1 properly in our body, what will happen? It will lead to deficiency and it will cause a disease called beriberi. Okay. So vitamin B1 causes beriberi. Next vitamin is vitamin B2, which is also called as riboflavin. So it will be absorbed, same thing, it will also absorbed in the duodenum and also in jejunum. Next, coming to activation. It will be activated by when riboflavin is converted into FMN and FAD, which will be utilized in your electron transport chain. So, what is the function? Oxidation reduction reaction and also electron transport chain. The storage, it will be stored in liver, heart, and also kidney. Its deficiency will cause erythroflavinosis. Next is your another vitamin which is known as niacin. Do you have any idea about the niacin? In your chemical, in your products which are using for the face, facial creams or serums, you will be using niacinamide. Okay. So vitamin B3, where it will be available instead of using creams to your faces, you can take good food like meat, fish, poultry, cereals, and legumes. And they can also be synthesized from the tryptophan. Okay. Coming to absorption, they will be absorbed from stomach and small intestine by passive diffusion and forms absorbed nicotinic acid and nicotinamide. Okay, they will be, how they will be transported? They will be circulated mainly as nicotinamide, transported into liver, muscle, brain, and the kidney. Next, coming to activation, nicotinamide will be converted into NAD plus, then NAD plus, NADP plus, which is high energy. Coming to storage, its storage is very limited and present mainly as NAD plus and NADP plus in tissue. I said that vitamin B or vitamin C cannot be stored for long time because they are water soluble vitamins. They will be left with the water more through your urine. Coming to excretion, excreted in urine as N-methyl nicotinamide. Okay, this deficiency can cause a pellagram, which is dermatitis, diarrhea. Mostly we are giving nicotinamide for the faces, right? So dermatitis. Dermatitis means skin disease. Okay. Next. So today we saw about fat soluble vitamins and also three of your water soluble vitamins. Okay. Students, did you understand?
Yes, ma'am.
Okay. If you have any doubts, you can just study and ask me in the next class. And there are remaining vitamins of the B also, and you have to study the C. See it, and if you have any doubt, you ask me in the next class. Thank you.
Thank you, ma'am.